Good morning, students. Welcome back to the class. We'll be discussing today's lectures about acquired immunity and adaptive immunity. From the past few classes, we already introduced a little bit about immunology, historical perspective, evolutionary perspective, and we briefly spoke about innate immunity and the cells involved and the mechanism innate immunity works to defend us from the bad. For example, in this slide, you see a distinction between innate and adaptive immunity. We already spoke briefly. So you see in this circle on the left, there are different kind of cells specific to the innate immunity. For example, dendritic cell, macrophages, eosinophils coming from hematopoietic stem cells. And at the same time, these cells we already discussed specific to the adaptive or acquired immunity, specific cells called B cells and T cells. And also we spoke in the previous class that there is both innate and acquired immunity work in cohort. For example, you see when the both circles merge here, there are specific cells called natural killer cells and the T cells. They have a specific communication or bridge functions between that. And also in the B and T cells, there, we will speak more about that in today's class. Coming on to the molecular mechanism for the innate immunity, we spoke that there is phagocytosis, there is inflammatory response that we discussed in the last class. But we already spoke in the previous classes in the introductory lectures about a specific cell called antigen presenting cell, APCs, and also how the APCs specifically give the information from the antigen to one of the cells called T cells or T lymphocytes that brings in part of the topic today, the acquired immunity. We will talk much more about details. The lecture is planned into the following ways. For example, all these concepts we will try to cover in today's lecture. We will start with acquired immunity. What is it about? Definition of acquired immunity. And then what are the cells involved in the acquired immunity? And then where the cells are produced, they are going through our blood and systems or different organs in the body, and how they are going to recognize the antigens through antigen presenting cells and other mechanism. We will talk, and specifically we'll speak about in detail on two of these processes of acquired immunity, that's called humoral and cell mediated immunity. And one important concept we will speak briefly is called clonal selection theory. And then a little bit briefly we can finish the lecture with a primary and secondary immune response that we were speaking already from past few classes. Okay, am I audible to everyone? If then it's okay, don't need to say it. So we are going back into the adaptive immune system, the definition wise. So adaptive or acquired immune system, also known as acquired immune system, is a subsystem of the immune system that is composed of specialized systemic cells and processes. It consists of specialized systemic cells and processes that eliminates the pathogen by preventing their growth and protect us from the disease. So the adaptive immune system is defined by this. Coming on to compared to innate and adaptive immunity possesses specific characteristic. If you take any example of any infection from bacteria, virus, or some autoimmune response, anything we will see, always there is a kind of response from adaptive immunity that follows a basic four characteristic features. The features is first one, antigenic specificity. How do you specifically recognize a foreign molecule, either antigen that consists of a specific chemical composition? That's first point, antigen specificity. And second point is the diversity. So you know that we are dealing with several pathogens, several viruses, bacteria, and some other pathogens. So we are encountered these pathogens in everyday life, and our immune system is the one protecting us. So there should be a kind of diversity that exists in the system, and if the diversity in terms of adaptive immune system, how it is established, we will speak. And also we know that there is an immunological memory. As immune system recognizes the pathogen for the first encounter, then the second encounter, it goes through an heightened response that there is a memory has been established within the cells. And also it recognizes cell from non-cell. So I will quickly glance through these points again, taking in each concept. For example, antigen specificity. We already know that antigen is recognized by a specific antibody. Antigen antibody is a fundamental of understanding that helps in the immune system. For example, an antigen is a toxin or any other foreign substance which induces an immune response in the body, especially production of antibodies. Antigen, it elicits an immune response that produces. That immune response and the antigen antibody reaction usually happens at the molecular and biochemical level, we will see in the second class at the unit 2 that talks more about antigen antibody interaction. We will talk about this, how is the molecular mechanism happens here. And also you should remember that antibodies possess highly efficient recognition potential. So there's kind of 6 antigens here, but there may be 6 trillion, 6 million, or 6,000, 60,000 antigens are there. The antibodies are highly specific to only one antigen, one antibody. For example, let's say antibody, there are antigens here, this color and this color, red and violet. An antigen specific to one of them can distinguish the antigen which differs, if it is a protein, which differs from a single amino acid residue. The antibody is for having the potential specificity that it can recognize the antigens having protein structure or protein molecule or amino acid residues that differ from a single amino acid residue. So the antibodies are highly specific. And also diversity. The immune system is capable of generating tremendous diversity in its recognition molecules, allowing it to recognize billions of unique structures or foreign antigens. See the amount here, the billions it can. How does it do it? So we don't go too much details into the antibody production, variability, and more into that. That will be part of the second class again. But for this lecture today, just look into the structure of the antibody, basic structure of the antibody. The antibody is a protein molecule that consists of heavy chain and light chain. Heavy chain Y-shaped, light shape is adjacent to that. And you see in the heavy chain and both light chain, there is a constant regions, colors here, green and blue. These regions in both these structure of the protein are constant. But there is a variable region here, the upper portion of that, where the antigen binding sites are present. It is highly variable and it is highly specific to a certain antigen. So you have billions of antigens. At the same way, you have billions of antibody that are produced in the cell through a specific molecular mechanism, and they will have a specificity, and each antibody in its own will have a variability of its production. So on immunological memory, again, we discuss first recognition process, first exposure, and the second exposure, it has a heightened immune reactivity, and it pertains to a lifelong encounter or long-term memory that establishes. And we already spoke several times about the self and non-self recognition. An antibody can recognize a self or a cell, endogenous cell to the body, having a self marker. It is happy to take the cell and doesn't do any immune response, doesn't need to destroy the cell. But if it takes an identifying a non-self, for example, antigen having another molecule where it is not happy about, it takes it as a non-self and then and it kills the cell or it destroys the cell. But just remember this process. For example, if there is a kind of flip over, an antibody being a self in the cell, carrying a non-self marker on the body, let's say these arrows come along in the self or endogenous cell, it gets confused, or it may feel as that in the endogenous cell may be threat and it may try to kill it. So that is one of the disease in autoimmune diseases, and also in the grafting reactions when you have a transplantation also it happens. And it should be remembered that there should be a distinct mechanism that protects endogenous cells, protects the body from killing the self, so that the body can function in terms of its growth, development, and other aspects of host pathogen defense mechanism. If it doesn't happen, if the immune system works in an aberrant way, our body is gone, we are going to be dead soon. So remember that self, non-self, and a different mechanism. We will speak in the lecture today further about these mechanisms. So again, I will take back now in the further few slides. What are the cells pertain to adaptive immunity? How the mechanism will happen into that molecular level, or in terms of a simplistic fundamental level, we will speak. And further in the lectures, we will talk more into advanced. So you know that adaptive immunity is compared of certain cells called lymphocytes that is produced into B lymphocyte and T lymphocytes. Again, T lymphocyte, T helper cell and T cytotoxic cells. Coming on to definition of the lymphocyte, what are lymphocytes? A lymphocyte is one of the subtype of a white blood cell, coming again from blood cells, hematopoietic stem cells in vertebrate immune system. Lymphocytes, there are several lymphocytes, but they have specific function. In innate immunity also, there is a special natural killer cell. It's also coming from the lymphocyte family, which induces cytotoxic innate immunity. But coming on to acquired immunity, T cells, they regulate cell-mediated cytotoxic adaptive immunity, B cells. Humoral, we will talk about that, what is cell-mediated humoral again in the next few slides. A B cell is again produced by the lymphocyte. And mostly these lymphocytes produced in a specific organ called lymph. That is, the name comes from lymphocyte. Basically, lymphocytes for acquired immunity, B and T cell, we will talk more about in the further slide. Coming on to the definition of the B cells, B cell is also known as B lymphocytes. They function in the humoral immunity and a component of adaptive immune system. Mainly the B cells work through secreting antibodies. You should remember that, highlight this information. The B cells are part of the adaptive immunity and they work through secreting antibodies. That is their functionality at the dead end. And there are two cells, T helper cells and T cytotoxic cells, TH and TC. T helper cell that provides help to other cells in the immune response by producing a specific signaling molecules called cytokines. T helper cells produce cytokines. The cytokines in turn will activate in turns T cells and B cells. Where they activate T cells is a kind called T cytotoxic cell. And again, B cells are also a kind of crosstalk happens where the T cell through cytokines elicits a response or activates a certain B cell. And the T cytotoxic cell, usually the T cytotoxic cell is produced when the body is under a kind of disease condition where the cancer is taking over the body and also an infection is happened through a viral infection. These two cases in a specific mechanism, T cytotoxic cells works, they kill cancer cells, and they also kill cells infected with the virus part. That when it comes to the mechanism, we will know, but we will stick to the fundamentals in the present class. So we spoke about lymph in the previous few slides. So we will talk about the lymphoid organs in the body. What are they? Where are they located? And what cells they produce? The cells involved in the immune response are effectively organized into tissues and organs. The cells, immune cells, which are different, B lymphocytes, T lymphocytes, and many other cells, basophils, monophils, and all other are divided into specific location in the body, the tissues and organs. These tissues and organs primarily divided into primary lymphoid organs, secondary lymphoid organs. Primary lymphoid organs, only two tissue systems or organs come up. That is, one is thymus located in the body here. Another one is a bone marrow. Bone marrow, hematopoietic stem cell divisions happens, cellular differentiation happens in the bone marrow. These two are primary lymphoid organs. And then secondary lymphoid organs are spleen, lymph nodes, and few other tissues that are as mentioned here. So why you call them primary and secondary? Because based on the origin of the cells, based on the functionality of the cells, which organ is important for the origin and production of the cell, which is important for the circulation and other aspects that you can discuss here. Primary lymphoid organs, thymus and bone marrow, are the major sites of lymphocyte development. That is the origin, development of the lymphocytes through a process called lymphopoiesis. Their function is to produce a repertoire of reactive cells that we will discuss in the few slides. And also the self-reacting cells, that means autoantigens, they protect that we will see in that. So basically they are important function, lymphogenesis, lymphopoiesis, that lymphocyte development. And secondary lymphoid organs, mainly their function is proliferation and maturation of the cells, not the production, but proliferation and maturation of the cells involved in that based on a specific signal, specific physiological condition in the body. And there is a cell-to-cell interaction, cytosine-cell interaction that we will see in the slides. So basically primary and secondary lymphoid organs based on the function, based on the what cells will do within that, they are divided into that. So an example here, you are looking into this slide. It's a kind of different lymphoid organs here, the bone marrow and fetal liver, thymus, and other tissues where the cells are circulating. For example, you take a condition of the B cell. B cell is produced through, again, blood cell stem cells, division of the blood cell, hematopoietic stem cells. At certain stage of the development, a certain cell of this blood stem cell of the blood will take into a B cell fate in the bone marrow. And then it will move into the lymphocytes, into blood lymphocytes that move in circulating into blood. And then it can go into the different tissues and organs through the blood, and it can reach to secondary lymphoid organs. Our blood circulatory system is the one carrying all these immune cells throughout the body, starting from the origin to the destination. So if you take an example of T cell is produced in the thymus, and thymus consists of, it also moves through the blood and through the tissue. So primary and secondary lymphoid organs, origin and distribution. And you just look into this specific structure here, importantly, in the secondary lymphoid tissues, we are talking about antigen presenting cells. What are these cells? They are specifically present in the secondary lymphoid tissues, not in the origin, because their sole purpose is to present an antigen to the primary lymphoid, primary lymphocytes. That is, so that's why they will be here, they will be not present in the origin cell. So what is an antigen presenting cell? Antigen presenting cell is a heterogeneous group of immune cells that mediate cellular immune response by processing and presenting antigens. Because we know that antigens, DAMs and PAMs we discussed in the previous class, they might have specific chemical structures, they might have been come through pathogen. So these antigen presenting cells help in processing them and presenting them in a form where the lymphocytes can recognize them. The lymphocytes may be again T cell, it may be mainly the T cells, the antigen presenting cells. And the classic APCs include dendritic cell, macrophages, Langerhans cells, and B cells are also part of the antigen presenting. Mainly the property of antigen presenting cell is that they express class 2 MHC molecule on the membrane. That is the marker, biomarker specifically for this antigen presenting cells. And they regulate a core stimulator signal in the activation of T helpers. So let's take an example here, how the antigen presenting cell interact with a specific T helper cell in a case here. So this slide is very important. You need to, we will take 1 or 2 minutes here or more than that to better understand this slide so that you will understand the concepts and other fundamentals of acquired immunity very well. So just go through the colors in this diagram, go through the arrows. I will guide you through here. Look into this chart. You are looking into a light gray color of the bars and then light yellow or orange and then red color bars. So first this bar represent antigen acquired from inside the dendritic cell. The cell here, the APC that is present in this scenario is called dendritic cell and it's talking to a T cell here through a receptor cell to cell communication. So antigen acquired from inside the dendritic cell. So this is the antigens that are in the green molecules. Let's say pathogen enters here through the dendritic cell and then it will undergo again endocytosis through phagosomes will come up. The phagosomes will break the cellular membrane or components of that. Along with that, whatever the chemical components of these molecules, the pathogen will be divided here into the endosome or phagosome. These are called antigens present from the dendritic cell from the pathogen by breaking the pathogen. And they will associate with the specific molecules called MHC protein, major histocompatibility complex proteins. They will go inside the phagosome. They will associate with the antigens here coming from the pathogen. And when the exocytosis happens, these MHC molecules, MHC2 molecules are associated with the antigens coming from this pathogen and move along into where the communication comes into the bridge between T cell and antigen. You see that green antigen, MHC molecule 2. Let's say a scenario antigen acquired from outside the dendritic cell. This is coming in and obtained from inside the dendritic cell. Let's say some antigens are already circulating in the body. Already it happened, a virus infected, some other infection, some specific chemical toxins are being produced. They are already circulating in the body. Then they also can enter into the system through dendritic cell. Endocytosis happens, phagosome. And again, they don't need to break or process anything. Immediately there is a chemistry and then another molecule MHC1 can go back and attach to that and then the complex forms, they can come back and present. So there is MHC1, MHC2, endogenous antigens and then specific from the pathogen. So it is clear the antigen presentation happens here and the ligand T cell receptor here, it can recognize the antigen here associated with the MHC marker. So this is the mechanism, it's a very important mechanism. Antigen is specific antigen in the body associating with MHC markers can bring an antigen. If this mechanism cannot exist, the T cells can go blind and they can try to recognize this chemistry of the antigens, but they cannot say it's a self, non-self unless they have these markers, MHC marker. That's why we have only specific cells, the 2, 3 or 4 cells that only become as APCs So there is a kind of control of the system representing this function. So you can read through the steps here. Step 1, 2, 3, and 4. You can understand what I have explained again in your free time. Thus, coming back to acquired immunity, there are two types of acquired immunity based on the molecules or chemical, in terms of the molecules involved. For example, antibodies or cytokines. So, for example, humoral immunity, you talk into humoral and cell mediated immunity, two kinds of acquired immunity. Coming on to humoral immunity, it is produced. Why the term is called humoral immunity? Because it produced in a body fluids are called humors. It happens in the body fluids, free floating body fluids. Body fluids which contain free floating molecules which are secreted antibodies, complement proteins, or certain antimicrobial peptides. So it acts through some proteins that flowing through the body, and they try to detect and defend the antigens, pathogens in the body. So that's why humoral immunity. And humoral immunity is also called antibody mediated immunity, specifically involving most of the antibody antigen reaction mechanism. Coming under cell mediated immunity, we are not talking about specific proteins or chemical components here. We are talking about directly cells. In cell mediated immunity, it doesn't involve antibodies. Please remember that it doesn't involve antibodies. Rather, cell mediated involves specific cells called phagocytes, antigen specific cytotoxic T lymphocytes, and release of cytokines that happens to them. So not only cytokines, a specific cell need to be involved in the response for cell mediated immunity. In humoral immunity, antibodies can do the job. So again, it's been clearly mentioned here, humoral cell mediated. For example, it's again B cell mediated humoral. The cell mediated is T cell, two types of T cells. And then we talked about here, mainly the T cell works through defending cancer and transplanted tissues. But you see the crosstalk here again. B cell can recognize an intact antigen, or antigens are specifically through APCs represented to the T cell, TH cell, and TH cell can send the response signal to B cell either through humoral, or it can send the signal to T cell through cell mediated cell to cell interaction, where they can activate a specific mechanism that gives rise to more and more production of antibodies that we will see how they are produced. Once a B cell is activated, it can be divided into, it goes into cell division process. It becomes into two daughters. One is a memory B cell, or two daughters, or it may be two fates. It takes two different fates. Certain population can become plasma cell. Certain population can become memory cells. What the plasma cell function, we will see into that. Same T cell, T memory cell, T active cell. But T helper cell go into this only one division, one fate. So humoral immunity, again, it acts through exogenous antigens, cell mediated, it targets endogenous antigens. It does not result in antibody production. Maybe a spelling mistake. So we already spoke that once the B lymphocyte is activated, it divides into two. And these plasma cells are short-lived and secrete high number of antibodies that are specific to a particular antigen. Please remember that we have cells in the body, B lymphocytes. They may be varied cells, but the structure is same, overall function is same, but they act on, but there is a kind of specificity is important here. So not all B lymphocytes will act on a specific virus infection. Not all B lymphocytes will work on a bacterial infection. They don't have commonality in terms of antigen recognition, but when you take a specific B lymphocyte, let's say 1000 B lymphocytes in a cell, all are same and they will have the same function to a particular antigen. So there is specificity, there is a clonal production that we will speak in the next slide. Plasma cells will secrete approximate 2000 antibody molecules per second. Please looking in the rate. Once activated a B lymphocyte, it goes into plasma cell and memory cell. The plasma cell can produce approximately 2000 antibodies per second, and they will flow into the bloodstream, whatever we discussed in the lymphoid organs, and they will stay up to 4 to 5 days in the system. Okay, remember this is very important. So how a specific B lymphocyte goes into a selection of specific antigen, and if there is an antigen molecules of 1000 are present, 1 lakh are present, the B lymphocyte that recognizes that specific antigen produces 1,000 or 10,000 or millions of B lymphocytes, they can go and kill the specific antigen molecule. How does it happen? It happens through a process called clonal selection. Clonal selection is nothing but cloning. So simple cloning of one B lymphocyte. You clone them into different populations having the same physical, chemical, and functionality. Clonal selection, it may be not only to B cell, it may be to T cell also. Clonal selection is a process proposed to explain how a single B cell or T cell that recognizes a specific antigen that enters the body, and then so a pre-existing cell pool will be there, because that we will talk later, but basically recognizes an antigen, then goes into an exponential way producing a clone of cell population for a specific antigen that eliminates the antigen further. How this process happens? This process happens again, whatever we summarized in the previous slides. Let's say an example of here, a dendritic cell, antigen presenting cell, recognizes a specific antigen in the body. Then it sends the signal to T helper cells only, right? You know that T helper cell only. And there is a specific interaction here. And then it produces a specific cytokine. And this will again go into memory TC cells, because it needs to remember, it needs to make immunological memory. It goes one way to become memory cells. In another way, it produces cytokines. Again, remember here, the cytokines are again specific to the antibody. Only one antibody. You see, in body, there will be a pool of antibodies, again, existing pool of antibodies. Let's say 4 here, it may be 40,000, 4 million may be present in the body. But a specific response to one antigen, it can elicited by only one antibody, one B lymphocyte carrying a specific antibody to that. So these cytokines are capable of recognizing that antibody to cell interaction, ligand receptor interaction. They pick that antibody only, and they tell the antibody, now you have to start. Go ahead, start producing more antibodies for this specific antigen. Then through this interaction, T cell and B cell interaction, the specific B cell is recognized. It allowed to go into cellular differentiation, cellular division, and it becomes a plasma cell, memory cells. But most of the population goes into plasma cell, and the plasma cell exponentially they divide, making 2000% per second antibody specific to the antigen, or it may increase depending on the demand, how we can go and defend the antigen or kill the system. So that's the clonal selection process, selection theory, and how it happens. So pool of antibodies already present in the body, but only why one specific elicit response comes up, because there is a specific recognition, there is a specific switch that happens. Remember that only the B cell that produces specific antibodies to the antigens which are activated by cytokines, not the T cell, not the APC, not the other cell. Only the B cell produces the lymphocytes. The antibody is called immunoglobulins. So pathogens typically contain multiple distinct antigenic fragments. You're talking about a pathogen, virus, or bacteria entered into the system. You're not talking about only one antigen, one chemical molecule released into the body. You're talking about several chemical components, several having distinct chemical components of the antigen. It may vary from in number. So basically, there should be a polyclonal activation system. So not only you're acting with one antigen, one chemical property. A single pathogen produce or stimulate several T and B lymphocytes to produce a specific antibody. Remember that. You talk one-to-one system, one-to-one clone selection in the previous slide, but you look into a system that work in a network, and then you talk about multiple antigens produced from single pathogen, then multiple T and B lymphocytes are produced, and a response comes through polyclonal activation. So again, coming back to the cells, this material has been provided from Kube Immunology book. Again, you talk into more in terms of functionality or structure of the cells. You look into the diagram explanation here. We talked about T and B cell, antigens, receptor antibodies on the B cell only. So a B cell will have about 10 power 5 molecules of membrane-bound antibody per cell. For example, if you take a specific B cell, you're looking into a production of 10 power 5 molecules of antibodies. Okay, and then all the antibody molecules on a given B cell have the same antigenic specificity and can interact directly with only one antigen. A specific B cell will have interaction to one antigen only. and you will have multiple repertoire of B cell that divides into other antigen. T cells, there again T helper cell, T cytotoxic cell. Remember that there is a specific signal again, peptide called CD4 positive cells. This is the molecule we will talk further about. This is CD8 positive cells. They have specific molecular biomarkers on the body, on the membranes. CD4 positive are T helper cells, and CD8 positive cells are T cytotoxic cells. And their number changes here, their interaction changes here. You can read through here. So we spoke in the present class, to summarize whatever we have spoken here. We spoke about what is an acquired and adaptive immunity, what are the cells involved in that, where are the cells produced and where they are circulating in the body, and how they recognize a specific antigen or multiple antigens producing for the pathogen, and how they can go into a specific process of clonal propagation through a clonal selection theory that we talked about in the present class. And we also spoke about antigen presenting cell, interaction of T and B cells, how, for example, cell to cell interaction, cell to cytokine interaction, ligand receptor interaction, how it works. And in several classes, we spoke about this concept of primary and secondary antibody responses. Again, we will brief up a little bit more on that. And now you might understand what is the primary response, what is the secondary response in a better way, rather than understanding only looking at the graph, some lag period, how the graph pitches up. But you take the same graph, same understanding and you go back with the lecture today, you look into that, why there is a lag period here, why there is a shortened lag period here in secondary response. You can relate it very nicely, you can relate that. For example, look in that, in the first response of an antigen, primary response, a novel antigen in the body, the lag period may be from days to weeks or sometimes months, because it has to go through a recognition process, it has to go to a production process, where whatever we discussed in the previous slide, a specific B cell has to be elicited, specific B cell has to divide, it goes into lymphopoiesis, to produce more lymphocytes or more antibodies to that. Whether it is able to produce or not, we don't know that. It's in the initial primary factory starting one new product at the first time. It takes a lot of glitches, you go into a kind of beta version. It will be tested many times. It may not work perfectly at times. So the lag period is more, but our immune system works through it. It works through it, lag period, then it produces antibody and the plateau will be there. What is the antibody specific that produced here? We will talk about in the further chapter 2, what is IgM, IgG. But at certain stage, it goes into plateau and the memory gets stored into a kind of memory cells, B memory cells that recognizes that and memorizes. And secondary response, again, it will take 3 to 4 days because memory cells will be there. It doesn't take months, but again, it takes a response in the recognition process. It takes some time, but the production will not take much time. Lymphopoiesis will be very heightened response. Total antibody production goes into higher value, and again, it peaks up, response will be high. So basically, you can summarize again in the text. Primary immune response, the first exposure, activated B cells are differentiating into plasma cells in the lag phase 2 to 3, and it takes months. The amount of antibody produced is relatively low. Over time, antibody level declines to the point where it may not be detectable at all. So what is IgG? We will talk further. And secondary immune response, the second dose of the same antigen is given days or even years later. An accelerated secondary or it's also called as anamnestic immune response occurs. The lag phase is usually short due to presence of memory cells. The amount of antibody produced is good high, and also specificity is also high. Antibody levels tend to remain high for longer time. You can detect longer time in the body. So that is primary and secondary immune response. You can look into that. You can relate to this point of clonal selection theory, specific cell-to-cell interaction. You can understand in a better way. So this is one of the chart produced from Encyclopedia Britannica. You can go through on the process of clonal selection theory here. I don't need to explain that in the class. I already explained in another graph, but you can read through the points. You can again understand the basic. Okay. So that brings back to the summary of the lecture for today. I will briefly stop the recording here. Okay, and then we will speak further again.